Hi everyone, welcome back once again to this educational channel on biology. In this lesson, we are going to study a topic from Form 4, and that is Chapter 2, Cell Biology and Organization. The subtopic is 2.1, Cell Structure and Function, and this is Part 2. This is the second video on cell structure and function. So join me as we take a tour of the cell to look inside and see what the interesting organelles and cellular components are, and also what types of cells have more of these organelles in order to carry out their functions well. So we are going to discuss the interesting aspects such as the structure and the function of these wonderful organelles in the cell that helps the cell to live and function efficiently. The learning standards for this lesson are as follows. 2.1 Cell Structure and Function, video number 2 or part 2. Firstly, we must be able to analyze the components of animal and plant cells as seen on the micrographs, which are the photographs of the enlarged components. Then state the main functions of components of animal and plant cells as seen on the micrographs. We will also be discussing the structures of the components or organelles such as the nucleus, Golgi Apparatus, lysosomes, and so forth. And then, apart from the structures, we'll discuss their functions. Then we are going to compare and contrast the components of animal and plant cells by discussing the similarities and the differences between animal cells and plant cells. Right, so overall, these are the structures that are found in the animal cell, such as the nucleus that contain the nuclear membrane and the nucleolus. Then you have the rough endoplasmic reticulum, the smooth endoplasmic reticulum, centrioles, mitochondrion, plasma membrane, cytoplasm, lysosomes, Golgi apparatus, and ribosomes. So we'll be discussing the structures and functions of all these structures in the next few slides. Okay, so this is an animal cell. Let's look at the plant cell. Right, so this is a three-dimensional diagram of a plant cell. Now, plant cells have similar organelles and cellular components compared to animal cells, such as nucleus, ribosome, endoplasmic reticulum, the rough ER and the smooth ER, Golgi apparatus, and mitochondria. However, there are three cellular components in the plant cell that are not found in animal cells, which are the large central vacuole, that is a permanent vacuole for matured plant cells, chloroplast, and cell wall. So we'll discuss this in more detail afterwards. Now here are the 2D diagrams for animal cell and also one for plant cell, the structure of animal cells and plant cells. Now this diagram was shown in the previous video, and it's important for us to know how to draw and recognize the 2D diagrams for Golgi apparatus, mitochondrion, rough endoplasmic reticulum, and centrioles, okay, and so forth. So if you have not copied the diagram, you can copy from here, and we'll be discussing the functions of the components later on. So here's the structure of the plant cell that you can copy if you have not done so. And here we see the same components as for the animal cell, except for the cell wall, chloroplast, and the big vacuole that's only found in the plant cell, but not in the animal cell. All right. A cell and a city. Now, what do these two have in common, the cell and the city? So a cell is like a tiny city. Now, in a cell, we have cellular components such as the organelles. For example, the nucleus, mitochondrion, and the endoplasmic reticulum, which carry out specific functions to help the cell survive. Likewise, in the city, we have workers, factories, places, and parts of the city which have similar functions or jobs compared to the organelles that help the city to develop and also to thrive. So as we take a tour of the cell, to find out more about its interesting cellular components, including the organelles, let us compare it with the workers' factories and parts of the city in order to understand the functions of the organelles and cellular components more clearly. So let's begin. So first, let us start off with the plasma membrane, which is the outer membrane that surrounds the cell. It forms the boundary around the cell. So when we look at the plasma membrane here, it seems like just a simple line, okay? But actually, if we were to analyze the structure more closely, it is not that simple because it is made up of mainly two types of molecules, okay? Proteins and the phospholipid molecules, which form two layers here. So we call it phospholipid bilayer. Right, we'll be studying more about this, the structure of the plasma membrane in chapter 3. Okay, so here we just need to know that the plasma membrane is made up of proteins or protein molecules like these, all right, which acts as the gateway to allow certain substances to move into the cell. And then also it is made up of phospholipids, which is a type of molecule that contains phosphorus and lipids in it. Okay, so this is the phospholipid bilayer. Now, so the plasma membrane is like a thin and elastic film that is partially permeable. Okay, so this is the important word that you have to know. Partially permeable or semi-permeable means that it only allows the free movement of certain smaller molecules across it, like glucose and water. Small molecules that can move through the pores of the plasma membrane. But it prevents and limits the movements of other substances such as the bigger molecules, like starch, okay, or sucrose. So starch cannot move through the plasma membrane. So if it's outside the cell, it cannot diffuse into the cell through the plasma membrane, whereas glucose can. So that's why we say that the plasma membrane is only partially permeable, not fully permeable. Okay, because only certain smaller molecules can move across it, whereas the bigger molecules cannot move across it. Now, so the functions of the plasma membrane, there are three here. The one that I highlight is the most important one that you can write down as the main point. If you're asked to give one function of the plasma membrane, okay, in the exam, then you can write it controls the movement of substances into and out of the cell. Okay, now let's start with the first one, functions. So the plasma membrane separates the contents of the cell here from the external environment, right? And B, it controls the movement of substances into and out of the cell. For example, glucose can diffuse into the cell through the plasma membrane, whereas starch cannot. Okay? Now, oxygen can diffuse into the cell also. And carbon dioxide can diffuse out of the cell through the plasma membrane. So C, another function of it is it allows the exchange of nutrients, respiratory gases, and waste materials between the cell and surroundings. For example, respiratory gases here are oxygen and carbon dioxide. Okay, so oxygen is used in cellular respiration, and it can diffuse into the cell from the surrounding fluid that's outside the cell. Oxygen diffuses into the cell for cellular respiration, and carbon dioxide that is formed through cellular respiration can diffuse out of the cell. Okay, so in that way, the plasma membrane allows the exchange of respiratory gases between the cell, the internal parts of the cell, and the surroundings. Okay, and for nutrients, glucose can diffuse into the cell, all right, through the plasma membrane. Right, a cell compared to a city. The plasma membrane of the cell can be likened to the city walls and gates, okay, and the city walls that surround the city. Then the city also allows only certain people to enter it, and it can shut the doors and the gates to prevent the entry of enemies, right, as in the olden days. Likewise, the plasma membrane is involved in regulating the substances that move in and out of the cell. Number 2, cytoplasm. Now the cytoplasm is the medium inside the cell. It is a jelly-like medium that is bounded by the plasma membrane. So it contains components, cell components or organelles, such as the mitochondrion, lysosomes, centrioles, Golgi Apparatus, and so forth. And these organelles are suspended in it. That means they are floating in the cytoplasm. All right. So apart from the organelles or cellular components in the cytoplasm, what else is found in the cytoplasm? Now the cytoplasm also contains substances, all right, in it, such as organic compounds, for example, proteins, lipids, and carbohydrates. These are food substances. So the word organic here means compounds that contain the element carbon, right? And the compounds contain the bond between carbon atoms. Okay. Then inorganic compounds are those compounds that do not contain the element or the carbon atoms, right? Such as the potassium ions. These are dissolved in the cytoplasm, right? So what is the function of the cytoplasm? The cytoplasm is important because it acts as a medium, okay, the location for biochemical reactions in the cells. For example, some reactions involving cellular respiration occur in the cytoplasm, okay, whereas other reactions involving cellular respiration occur in the mitochondria, all right? So the cytoplasm is the medium for biochemical reactions in cells. So if we were to compare a cell to a city, the cytoplasm in the cell can be likened to the city grounds, okay? Can be likened to the ground in the city where buildings are built on it, all right? And these city grounds may also include the sand, the soil in the city on the land, or the land in the city, okay, where the buildings are found, okay. Number 3, the nucleus. So the nucleus is the largest component in the cell. It's spherical or round in shape, and is enclosed in a nuclear membrane, right? The pink structure here, with pores or holes to allow substances to move in and out of it. So in the nucleus, we have the nucleolus, which is a dark black spot, all right? Here is the 2D diagram, nucleolus. Also in the nucleus, we have chromosomes, which are tiny thread-like structures that carry genetic material, all right? And is involved in inheritance or pewarisan, meaning that these chromosomes can be passed down from parent to child, all right, through the sperm and the ovum. So apart from chromosomes, there's the nucleoplasm, which is the medium inside the nucleus. Just as you have cytoplasm, okay, outside the nucleus in the cell, then you have nucleoplasm in the nucleus. Now, what are the functions of the nucleus? So the nucleus is the big boss, okay, in the cell. It controls all the cellular activities in the cell, and apart from that, it has chromosomes that contain a molecule called deoxyribonucleic acid, an important molecule. In short, we call it DNA. D, okay, here D, N, and then A. So this molecule determines the cell's characteristics and metabolic functions. How does it do it? Because DNA is actually the molecule that carries genetic instructions, or the genes, okay, that tells the cell what to do. And it is involved in giving the instructions for growth and functioning of living organisms. All right. So we'll be studying more of this important molecule later on. Okay, not to worry. Just remember that the chromosomes contain deoxyribonucleic acid, and that this DNA contains genes or genetic information or genetic instructions, which determines the cell's characteristics, even the organism's characteristics, and also the metabolic functions in the cell. Thus, the nucleus of the cell can be compared to the administrative center of a city, or the mayor of a city, which is the boss or the chief executive officer of the city that oversees and directs activities of the city's main departments like police, fire, education, etc. Right? So because the nucleus controls all the activities in the cell. So we can liken it to the mayor, all right, of the city. Number 4, the mitochondrion. Plural form is mitochondria. Many mitochondria. One mitochondrion, many mitochondria. Structure. A mitochondrion is rod-shaped or spherical, all right? So it can be longish in shape like this. Now the important thing is that it consists of 2 layers of membranes, the smooth outer membrane and the folded inner membrane. All right, now in the 2D picture you can see the smooth outer membrane and the folded inner membrane. So for the inner membrane, it is folded to form folds called crista. A crista is a fold of the inner membrane, and the plural form is cristae, C-R-I-S-T-A, add an E there, T-A-E, cristae. That's the plural form. So we can say that the mitochondrion has double membranes, 2 membranes, in which the inner membrane is folded. Okay, so what's the use of the folds? Okay, so the folded inner membrane helps to increase the total surface area of the inner membrane, right? Make the surface area larger as compared to a smooth inner membrane. So the folds increase the total surface area so that more energy in the form of stored in the ATP molecules can be generated through cellular respiration, because cellular respiration occurs in the mitochondrion, and some of these reactions occur on the surface of the inner membrane, right? So the larger the surface area of the inner membrane, then more energy can be produced through the increase in the number of chemical reactions on the surface of the inner membrane. So this is to make sure that more energy is produced through cellular respiration in the mitochondria. Now, continuing with the structure, the mitochondrion contains enzymes, respiratory enzymes that play a role in cellular respiration. Okay, these enzymes catalyze or speed up the rate of reaction in cellular respiration. All right. So what is our equation for cellular respiration? Glucose plus oxygen produces water, energy, and carbon dioxide. This is called the word equation, and our short form or acronym is GOWAC. G for glucose, O for oxygen, W for water, E for energy, and C for carbon dioxide. So this is a good short form to remember for the equation for cellular respiration. Okay, take note of this. Now, the function of the mitochondria, it is the powerhouse of the cell because it generates energy through the oxidation of glucose, okay, where glucose reacts with oxygen during cellular respiration. And then energy is released, all right? But this energy is not used straight away. It is stored in a molecule called ATP or adenosine triphosphate, ATP, in cells. So ATP can be called an immediate energy store. So when we need energy, let's say for muscle contraction, in order to move our hands and legs, we need to contract our muscles. And to contract muscles, we need energy. Then the ATP in the cells of the muscles will break down. When it breaks down, it will release the energy that actually comes from cellular respiration, actually, but which was stored in the ATP. So this energy will be released for muscle contraction and for other cellular activities like cell division, transmission of nerve impulses, and so forth. All right. So a summary is like that. The summary is like this. Cellular respiration will generate energy, right? So this occurs in the mitochondria. But this energy is not used immediately. It is first stored in the ATP molecules in cells. So muscle cells have a lot of these ATP molecules because they need to contract. So they need to get that energy store ready first. Okay? Then when the cells need energies, for example, for muscle contraction, the ATP molecules in the cell will break down and release that energy for muscle contraction and other cellular activities. All right, so ATP is considered an immediate energy store or just an energy store. Thus, if we compare a cell to a city, the mitochondria in the cell can be compared to the power station generating electrical energy in the outskirts of the city. So this electrical energy is generated for use by households and to power devices and so forth. So the mitochondrion, likewise in the cell, can be considered the powerhouse of the cell because it produces energy for the cellular activities to be carried out. Now, cellular component number 5 are the ribosomes. Ribosomes are small, compact, spherical granules. So from far we can see that ribosomes look like round dots, and they consist of proteins and ribonucleic acid, RNA, ribonucleic acid. So the ribosomes are made of these molecules, proteins and ribonucleic acid. Now, what is ribonucleic acid? Ribonucleic acid is a chemical substance or nucleic acid present in cells. So this RNA and DNA will be studied later on in another chapter, so don't worry about that. Now let's get the location. Ribosomes are present either on the surface of the rough endoplasmic reticulum, the green structure here, so they can be attached to the rough endoplasmic reticulum, or they can exist freely in the cytoplasm, floating in the cytoplasm. Okay, so the function of ribosomes is very important. It is the site or the location for protein synthesis or production of proteins. Synthesis means production. All right, now this is the 2D diagram showing you the rough endoplasmic reticulum and the ribosomes attached on it. Okay, so ribosomes produce proteins which are later used to form the enzymes, antibodies, hormones, and other substances made of proteins. Okay, so it's very important to the cell. Now, if you were to enlarge one ribosome, you'll see that actually it is not round. It has two subunits, the large subunit at the top and a smaller subunit at the bottom. So how do ribosomes make these proteins? How do ribosomes make proteins? They do so by using a genetic code or genetic instructions that come from the nucleus, that is sent by the nucleus. Okay, now we'll be studying more about ribosomes in another chapter when we talk about the synthesis of proteins in greater detail. If we compare a cell to a city, the ribosomes in the cell can be likened to small food factories that produce simple foodstuff like milk or cocoa powder, right? Because ribosomes produce proteins which later are sent to the Golgi apparatus to be modified into enzymes and so forth, right? So likewise, the small food factories will produce a simple foodstuff which are later sent to the bigger factories to manufacture more complex foodstuff. Okay, like milk and cocoa powder produced by the small factories will be later sent to the bigger factories to produce complex foodstuff like chocolates, cheese, and so forth. Cellular component number 6 is the endoplasmic reticulum, right? Now, reticulum here means a network, and endoplasmic could mean inside the cytoplasm. Endo means inside, plasmic refers to the cytoplasm. Inside the cytoplasm, you have a network of structures. Okay, so the endoplasmic reticulum generally consists of a system of interconnected, folded, flattened sacs like this one, right? Flattened bags, continuous with the nuclear membrane. So they are joined to the nuclear membrane that surrounds the internal parts of the nucleus. That's part of the nucleus. Now, there are two types of endoplasmic reticulum, ER, namely the rough endoplasmic reticulum, because it has ribosomes attached on its surface. So if you were able to feel it, it would feel a bit rough, you know, because of the ribosomes on it. And then there is the smooth endoplasmic reticulum. Is called the smooth endoplasmic reticulum because it does not have ribosomes attached on it, right? So this is the rough endoplasmic reticulum. In short, we call it RER, and this one is the smooth endoplasmic reticulum, called the SER, in short. But in exam, as I said, you have to write down the full name, spell it all out. So here we can see the difference in the structure of the rough ER and the smooth ER. The rough ER is like flattened bags, okay, but the smooth ER is more like vertical tubes that are interconnected. All right, so there's a slight difference. And then if you look at the 2D pictures of it, rough ER, you have to draw the ribosomes on it, and for smooth ER, there are no ribosomes on it. Okay, now the function. So generally, the function of the endoplasmic reticulum is that it is the transport system within the cell. Why? Because it transports various substances like proteins, lipids, and so forth, generally. But we are going to discuss the specific function of rough endoplasmic reticulum and smooth endoplasmic reticulum in the next slide. So generally, the endoplasmic reticulum is the transport system within the cell, and it provides a wide surface. Okay, now I can see the surface is very wide because of the flattened folded shape. So a wide surface for what? Enzyme attachment and also biochemical reactions on its surface, right? Now let's discuss the specific functions of the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So we start with the rough endoplasmic reticulum. The function of the rough endoplasmic reticulum is that it transports proteins, say the word proteins, made by ribosomes. So these tiny ribosomes on the surface of the rough endoplasmic reticulum will first produce or synthesize proteins, and then the proteins will go into the space inside the rough ER and is transported in the space in the rough ER until it reaches the end of it, where after that the protein is enclosed in transport vesicles that will move to another organelle called the Golgi Apparatus to be modified. The proteins need to be modified to form enzymes and so forth. Okay, so the rough ER transports proteins made by ribosomes, and it also encloses the proteins in transport vesicles, which look like just round bags like that. And the transport vesicles will move to the Golgi apparatus and empty its contents, that means the proteins, into the Golgi apparatus to be modified. All right. So remember, rough ER has something to do with the transport, not synthesis, but transport of proteins. Let's talk about the smooth endoplasmic reticulum, or SER. Now, this is the structure of the smooth endoplasmic reticulum. It looks like a series of tubes that are joined together, right? And this is the 2D picture of it. If you're asked to draw, then you draw like this. Draw the smooth ER like this. So what are the functions of the smooth ER? The smooth ER, or smooth endoplasmic reticulum, synthesizes and transports glycerol and lipids. Okay, unlike the rough ER that transports proteins. Okay, so smooth ER synthesizes the glycerol and lipids and then transports them, right? So it transports different substances from the rough ER. Okay, so imagine to remember this, you imagine a smooth ER. Lipids are, for example, oil is a lipid, right? So it's when you touch it, it has a smooth feeling. So remember, smooth ER synthesizes and transports lipids and glycerol. Now, the second important function of the smooth ER is that it carries out detoxification of drugs and metabolic byproducts. Okay, so detoxification means the removal of toxic or poisonous substances from an organism, right? So sometimes a person may eat something unknowingly that is toxic, right? So it will be sent to organs like the liver, where the detoxification of the substance will occur to make it not poisonous to the body, all right, or to neutralize the toxins. Now that's the job of the organ called the liver. So the liver has a lot of this smooth endoplasmic reticulum to carry out detoxification of poisonous substances. Okay. So if we are to compare a cell to a city, the rough endoplasmic reticulum and the smooth endoplasmic reticulum, okay, which together is called the endoplasmic reticulum, actually is like the transport system in a city, which are the roads, railways, and rivers that transport substances from one place to another. Okay, so the rough and smooth ER is like the transport system in the cell. Next, we have number 7, Golgi Apparatus. Now, the word Golgi actually comes from the name of an Italian physician and histologist called Camillo Golgi, who discovered the Golgi apparatus. He was awarded the Nobel Prize in Medicine in 1906, right? So when you write the word Golgi, the G must be a capital G because this is the name of a person, right? So the structure of the Golgi apparatus, it is a stack of parallel flattened sacs or bags. These are the flattened bags, okay? Enclosed by a single cell membrane, so it's only one membrane, and not double membrane. Now, new membrane is added at one end of the Golgi apparatus, and vesicles bud off from the other end. So what are vesicles? Now, this is the 2D structure that you can draw if you're asked to draw the Golgi apparatus, and these are the vesicles, right? So vesicles are just tiny sacs or bags that transport substances in the cell, all right? So examples are transport vesicle and secretory vesicles, which we will discuss later. Now, so the Golgi apparatus is very important. Now, you can see here the structure is like a C-shape, parallel C-shape structures, okay? And then with the vesicles at the side. So this is how you can recognize the Golgi apparatus compared to, if you compare them to the smooth endoplasmic reticulum. The Golgi apparatus, it has vesicles at the side and then C-shaped structures like this, okay? So it's different from the smooth endoplasmic reticulum. Now, function. Function is very important. It's very often tested in the exam. The Golgi apparatus processes, modifies, packs, and transports proteins, carbohydrates, and glycoproteins. Glycoproteins are a combination of carbohydrates and proteins. So the Golgi apparatus receives the proteins made by the ribosomes, okay? Then after that, it will process these proteins, modify it, that means to make some changes to the proteins so that the proteins become the enzymes, all right, or other products. Then it packs it in the secretory vesicles, all right, which then bud off from the Golgi apparatus and move to other places, okay? So if you ask the function of the Golgi apparatus, you can say it processes, modifies, and packs the proteins, all right, as one of the main functions. And secondly, it packages the enzymes that are formed from the modification of the proteins into the secretory vesicles, which then move away from the Golgi apparatus, and the secretory vesicles may go to the plasma membrane and secrete the enzymes out of the cell, right? So we discuss that in the next slide. Now, if we compare a cell to a city, then the Golgi apparatus can be likened to a big factory that processes and packages complex foodstuff or products, such as chocolate and cheese. Now, to understand this more, let us discuss how a digestive enzyme can be produced by the organelles in the cell, all right? So how are enzymes produced by the cell? So first we have the ribosomes, which are the tiny organelles on the rough endoplasmic reticulum. Ribosomes will synthesize the proteins first of all, and then the proteins are transported in the rough endoplasmic reticulum, okay, this purple structure here, After that, the rough endoplasmic reticulum will package the protein in a transport vesicle, which acts like a lorry, right, to transport the proteins to the Golgi apparatus. This one, okay, the blue structure is the Golgi apparatus. Now, why do the proteins need to be transported to Golgi apparatus? To be modified. Okay, that means the Golgi apparatus will make some changes to the proteins, and then they will then produce the enzymes, like digestive enzymes such as amylase or lipase, right? Then after that, these enzymes have to be packaged in secretory vesicles, which move to the plasma membrane here, where the enzymes are secreted. Secreted means they are released or discharged outside the cell, all right? And then these enzymes will then move to their destination, such as the small intestines, okay, where they will carry out digestion of food, all right? So this is the way that the enzymes are produced. And so pancreatic cells, that means cells in the pancreas, certain cells in the pancreas have a lot of this rough endoplasmic reticulum and Golgi apparatus, okay? In order to synthesize the enzymes, right, we need the rough endoplasmic reticulum with the ribosomes and the Golgi apparatus to synthesize the enzymes, and then also to secrete or release the enzymes to the outside of the cell. So this job is done by the Golgi apparatus because it will package the enzymes in secretory vesicles that move to the plasma membrane so that the enzymes like lipase and amylase can be secreted outside the cell. All right? So take note of this. Our pancreatic cells have a lot of rough ER and Golgi apparatus to synthesize and secrete a lot of digestive enzymes such as lipase, amylase, and also trypsin. Okay? And all these enzymes are proteins. So they start, the production of these enzymes start with the ribosome that synthesizes the proteins. And then the proteins are modified by the Golgi apparatus to form the digestive enzymes. Next, we have the centrioles. So centrioles are small cylindrical components that exist in pairs. There are two centrioles in each cell, and they're only found in animal cells, not in plant cells. Okay, so each centriole is made up of a complex arrangement of microtubules. Microtubules are very tiny tube-like structures. Okay, micro means tiny, and tubule is really also tiny tube. So a centriole is made up of a complex arrangement of microtubules, as you can see here. And this is the cross-section of the centriole. You can see all these circular parts are the microtubules. Okay, they are arranged in a complex pattern like this. So centrioles do not exist in plant cells. All right, please take note of that. And the function of centrioles, okay, the pair of centrioles, is that it is involved in cell division. It forms fibers called spindle fibers during cell division in order to pull the chromosomes apart from each other, right, during cell division in animal cells only. Okay, so memorize the function of centrioles. So we'll be discussing more about the function of centrioles when we study cell division. Okay, now this is the definition of centriole. Centrioles are cylindrical structures that are made up of protein tubes called microtubules, as you can see here. They look like a bundle of pencils. So 9 groups of 3 microtubules. 9 groups of these, called the triplet microtubules, are linked together to make the walls of the cylinder. So it looks like a cylinder that's made up of the tiny tubes. Okay, that's a centriole. Now, the comparison of a centriole, okay, in a cell with something in the city. So I have not found any suitable comparison yet. Okay, so we'll just leave it as it is without any comparison with the structures in the city. Next, we come to the waste disposal unit of the cell, the lysosome. So a lysosome is made up of a small spherical sac or bag enclosed in a single membrane. Okay, this is the red part. Here is the membrane. And it contains hydrolytic enzymes inside, as shown here. So hydrolytic enzymes are enzymes that can digest substances. All right, hydrolytic means to break down. It's able to break down substances. Now the functions. It hydrolyzes or digests or break down complex organic molecules like proteins, nucleic acids, and lipids in the cell. And it can get involved in digestion of these organic substances. Okay. Secondly, it can break down bacteria. Okay, for example, white blood cells have a lot of lysosomes to break down the bacteria that it has engulfed. Okay, and lysosomes can also digest or break down components of damaged cells, such as damaged mitochondria or old mitochondria, right? So it digests the bacteria and old organelles and then removes them, okay? So through that way, it removes them. So it digests and removes the waste inside the cell, right? That's why I say it is like the waste disposal unit. So here we can see the cross-section of a lysosome. It has only a single membrane, unlike the mitochondria or chloroplast. And then inside you have the hydrolytic enzymes, right, that help to hydrolyze or digest the organic molecules or bacteria or even damaged organelles. Now, sometimes the cell has damaged organelles. It needs to get rid of all these waste substances, these substances that are not needed. Okay, so the lysosomes will do their job to break down the damaged organelles and remove them from the cell. Right, so if we compare a cell to a city, the lysosomes in the cell can be likened to the waste disposal units in the city. Okay, for example, the disposal unit that collects the trash from the households, and also the workers that sweep and clean up the streets, right, every day. So in that way, the waste products are disposed of. All right. So that is something like the function of the lysosomes in the cell. Right, next, we have the cell wall. So the cell wall is the outermost layer of the plant cell. Now, structure. It is a strong and rigid outer layer made of cellulose fiber. Cellulose fiber is a tough fiber. For example, when you eat vegetables, sometimes you can find that the fibers are not easily broken up. Those are made of cellulose, right? So no matter how hard you chew your food, also you cannot break down the cellulose fiber sometimes. So that is the tough cellulose fiber. That's how tough it is. And the cell wall is fully permeable. That means it allows all the common substances to move through it. All right. Unlike the plasma membrane, which is semi-permeable. All right. So the plasma membrane is the membrane that is found below the cell wall. That's the next membrane. Okay, that is side by side with the cell wall. Okay, so so here the white line here can represent the plasma membrane. The green part is the cell wall, then the white line here is the plasma membrane on the inner part of the cell wall. Now the function of the cell wall is that it maintains the shape of the plant cell because it's tough and rigid, and it provides mechanical support to the plant cell in that way. Next, another organelle that's only found in the plant cell is the chloroplast, right? So the chloroplast is green in color, and structure is oval shaped. Its structure is oval shaped, and it consists of two layers of membrane, the outer membrane and the inner membrane, just like mitochondria, except that the inner membrane is not folded, whereas the inner membrane for mitochondrion is folded. Okay, so the inner membrane is also smooth and not folded. Then inside the chloroplast, there are green pigments or coloring substance called chlorophyll in the structures that look like stacks of coins. Okay, So let's see how to label it. Now, so this is the drawing that you should draw if you're asked to draw the structure of a chloroplast. You must draw that it has double membranes, outer membrane and inner membrane. And then inside, you can just imagine that it has structures that look like stacks of coins. So like each coin here, within inverted commas, or each circular structure here is called a thylakoid. Try and say this word, thylakoid. Okay, so a stack of thylakoid is called a granule. Remember, group. So a group of thylakoid is a granule, right? So then the liquid or the matrix in the cell is called the stroma, not stoma, stroma, right? So you must be able to label all these parts. So the color of the thylakoids are green because it contains the green pigment called chlorophyll. Okay, whereas in the stroma there is no chlorophyll. Now, what's the function of the chloroplast? The pigment chlorophyll in the chloroplast absorbs sunlight and converts it into chemical energy in the bonds of glucose during photosynthesis, right? So the chloroplast contains chlorophyll which can absorb sunlight, all right, to carry out photosynthesis. And then in the process of carrying out photosynthesis, it converts the light energy into chemical energy when glucose is formed through photosynthesis. According to the equation WEC-GO, water plus energy plus carbon dioxide produces glucose and oxygen. So when the glucose is formed, the bonds between the atoms in the glucose actually stores chemical energy. Okay, so if we want that energy, we can break down the bonds of glucose during cellular respiration to release the energy for cellular activities, right? So photosynthesis is just the opposite of respiration. And it needs the pigment chlorophyll in the chloroplast to absorb the sunlight for the photosynthesis to be carried out by the plant. And for light energy to be converted into chemical energy stored in the bonds of glucose. Now, if we were to compare a cell to a city, the chloroplast inside the cell can be likened to the solar panels, okay, and on the rooftops of buildings in the city that can absorb sunlight, because chloroplast contains chlorophyll that absorbs sunlight and converts it to chemical energy stored in the bonds of the food molecules. Vacuole. So a vacuole in a plant cell is actually a liquid-filled sac that contains the fluid called cell sap, right? So here's the vacuole, right, in the plant cell. It's a big one. And this is the 2D diagram. So we can see that the vacuole has a membrane called the tonoplast that surrounds the cell sap, right? So a young plant cell will have many small vacuoles, but for a mature plant cell, as shown here, it has only one large vacuole that is almost in the center of the cell, right? Whereas for animal cells may have vacuoles, but they are small and usually temporary. Okay. Now, what about the cell sap? What is found in the cell sap? So cell sap contains a lot of substances like water, organic acids, sugars, amino acids, enzymes, mineral salts, oxygen, carbon dioxide, and metabolic byproducts. This means that the vacuole stores all these substances in the cell sap, right? So you must be able to name at least 3 substances like water, sugars, and amino acids that are found in the cell sap. Now, what is the function of the vacuole? So for plant cells, when water is absorbed into the vacuole from the external environment, the vacuole will expand and then it will press against the cell wall, okay, of the cell. So this causes turgor pressure to occur, right, which is the pressure of the vacuole on the cell walls. And this makes the cell become turgid, that means firm and hard. So it will give support to the whole cell. So if many of the plant cells in a plant are turgid, then the cell turgidity will give support not only to the cells but also to the whole plant. So the plant will If the cells in the plant are turgid, then the plant will be able to stand upright and firm. Okay, not withered, right? So, secondly, in unicellular organisms like amoeba and paramecium species, right, there are contractile vacuoles which play a different role. So contractile vacuole, from the word contract, they can contract. Contractile vacuole can contract during a process called osmoregulation to control the amount of water in the cell. So these organisms live in fresh water, and water is always diffusing into their cells by osmosis, by a process called osmosis, which we'll study later. Then this water will move into the contractile vacuole, and the contractile vacuole will expand, and when it reaches maximum size, it will contract to excrete that excess water out of the cell so that the cell will not burst. All right, so this process is called osmoregulation. We'll be studying in the next subtopic. Okay, so not to worry about that. Now, if we were to compare a cell to a city, the vacuole in the cell that stores water and sugars can be likened to a reservoir in the outskirts of the city that stores water, right? But the vacuole stores more than water. It stores sugars, amino acids, mineral salts, and so forth. Let's look at this common question on comparison. Compare and contrast between the components of an animal cell and a plant cell, 10 marks. So compare and contrast questions are very common in biology, and we must be aware that when you are asked to compare and contrast, we have to state both the similarities and the differences between the two objects. For example, between an animal cell and a plant cell. What are the similarities and what are the differences? Okay, so let's start with the similarities. For similarities, you can begin with both. Both cells, both animal and plant cells, contain here the organelles that are common in both cells are the nucleus, cytoplasm, plasma membrane, Golgi apparatus, mitochondria, endoplasmic reticulum, and ribosomes. Okay. Now, how about the differences? So animal cells do not have a lot of things, don't have some organelles that are found in the plant cells, okay, and do not have certain components that are found in plant cells. For example, now let's begin the first one. From the aspect of shape, an animal cell does not have a fixed shape, as we can see when we look at the cheek cells of a human being. But for plant cells, usually they have a fixed shape. For example, the epidermal cells of the onion, which you have observed under the microscope. It has a fixed shape, right? Now then, animal cells do not have cell walls, but plant cells have cell walls. An animal cell does not have chloroplasts, which are involved in photosynthesis to produce organic food substances. But plant cells have chloroplasts, so they can synthesize their own organic food substances like sugars, like glucose. An animal cell usually does not have vacuoles. If present, also the vacuoles are small. But for plant cells, it usually has a large vacuole near to the center of the cell, right? Most plant cells have large vacuoles. Now, an animal cell stores carbohydrates in the form of glycogen, not starch. But plant cells store carbohydrates in the form of starch. Okay. Now, both glycogen and starch are called polysaccharides. So they are complex carbohydrates, but their structure is different. Okay. Then we have centrioles. So this is the one organelle found in animal cells that is not found in plant cell. Okay. Animal cells have centrioles that are involved in cell division to form the spindle fibers, right, in cell division. But plant cells, they do not have centrioles. They can still carry out cell division without centrioles. Now finally, how about lysosomes? So some students ask me, do plant cells have lysosomes? Now the answer is that lysosomes are mainly found in animal cells. All right, mainly found in animal cells and rarely found in plant cells. So there may be plant cells with lysosomes. Okay, we do not know, but they are rarely found in plant cells. Okay, So they are generally more common in animal cells. Right, that's all for this lesson. Thanks for watching. Do share, like, and subscribe. Goodbye for now.